Hi, I'm Tali, and I will present my thesis project under the supervision of Dr. Yaara Yeshurun. It's about neural synchronization as a function of engagement with the narrative. The degree of engagement to a narrative varies across people and depends on the situation. For example, two parents reading to their daughter Little Red Riding Hood is likely, whereas without their daughter it is less plausible. In the present study, we sought to find the neural correlates of engagement to a narrative based on the differences of engagement ratings between participants. We hypothesized that the engagement ratings would be associated with the neural responses, specifically in the default mode network, DMN. This network is known to be involved in processing narratives. We reanalyzed data collected for a previous study. 25 participants listened to a story while undergoing an fMRI scan. After the scan, they answered a questionnaire, which assessed the degree of their overall engagement. The data processing diverged into the voxel-by-voxel approach and the parcellation approach based on Yells and Fans maps. In every approach, we conducted 3 methods of analysis. Inter-subject correlation, the ISC method, is meant to find correlated parcels or voxels across all participants. Finding such a correlation indicates that they are activated similarly across most participants. It is safe to assume that these regions have to do with the processing of the stimulus in light of the fact that this stimulus is a synchronizing factor of all the participants. Inter-subject representational similarity analysis, the ISRSA, is a method that aims to bridge neural and behavioral activities. This analysis illuminates brain regions that are more synchronized among more engaged participants. And finally, intersubject functional connectivity, the ISFC method, finds distinct correlated regions among participants. This is a way of finding synchronized functional connectivity, or in other words, a less noisy functional connectivity. These are the results of the ISC analysis. On the left side is the parcellation approach, and on the right side is the voxel-by-voxel approach. In both approaches, the auditory cortex, DMN areas, and linguistic areas were correlated across all participants. This is a typical result for ISC analysis of naturalistic stimuli. ISRSA, based on the parcellation approach, revealed a correlation between increased engagement with the story and increased neural synchronization within the following: regions of the frontal cortex, the posterior cingulate cortex, and the temporal pole. The voxel-by-voxel approach adds to this list the areas around the bilateral caudate nucleus. This is not a typical finding because this area is in the white matter, known to have no blood flow in it. This is the intersubject functional connectivity results for the parcellation approach. Later on, we will view the voxel-by-voxel approach. Yell's parcellation map can be divided into networks. Their names and abbreviations are on the bottom left. The participants were divided into 3 groups defined by their engagement ratings. The least engaged group is on the left, and the most engaged group is on the right. All in all, it seems like synchronized functional connectivity of the brain increased as the engagement of the participants increased. This is an illustration of the connectivity between neural networks. Specifically, the positive connectivity between the DMN and the frontoparietal network, FPN, increased, as well as the positive connectivity between the DMN and the ventral attention network, VAN. These correlations are still not corrected for multiple comparisons. It is also evident that the synchronized functional connectivity within the DMN was the highest among the participants who were the most engaged. An interesting finding is that the least engaged participants displayed a higher synchronized functional connectivity than intermediate engaged participants. The synchronized functional connectivity within the FPN slightly increased as the participants were more engaged, although this finding has not yet been corrected for multiple comparisons. These are the results of the voxel-by-voxel approach for the intersubject functional connectivity analysis. They too support the claim that the synchronized functional connectivity increased as the engagement of the participants increased. Our findings suggest that engagement to a narrative may shape the response of the DMN and attention regions. As the engagement increased, the connectivity increased within the DMN, between the DMN and FPN, and between the DMN and VAN. Our findings also show a stable connectivity within the FPN, if not a slight increase. While the DMN was classically referred to as the task-negative network, as it is inactive while completing a task, the FPN was classically referred to as a task-positive network, as it is usually active while completing a task. Our findings accumulate to recent ones that imply that these networks sometimes do work together and are not mutually exclusive. Naturalistic stimuli are gaining more and more popularity, and it is important to make sure that they meet the needs of the experiment. There was a difference between the ISC map and the ISRSA map. This difference indicates that the brain regions that were more synchronized as engagement rates were higher, as shown in the ISRSA map, did not show synchronization among all participants, as shown in the ISC map. This implies that the usage of naturalistic stimuli should take into account the engagement involved. Thank you for listening, and this is my email for further discussion.